Hi everyone, this is the DNA replication notes. This is the overview image of what's going on. So as you can see, we have a pre-existing DNA strand that is starting to unwind, and new bases are starting to be added to this DNA. So the unwinding enzyme is called helicase, probably because it is unwinding and unzipping the helix or double helix, as you can see. It unwinds it between the nitrogenous bases. Then we have another enzyme called DNA polymerase. DNA polymerase is going to find free-floating nucleotides, which are our subunits of DNA found in the nucleus, and it's going to add the bases using the base pairing rules, A with T, G with C. So, a helicase is an enzyme. And an enzyme is a protein that acts as a biological catalyst, so it helps speed up reactions. So helicase is a catalyst because it is going to help DNA replicate faster. And it does that by breaking those hydrogen bonds between each strand of each side of the DNA. And then we can add in bases using DNA polymerase. So DNA polymerase is the next enzyme here. So once again, it's another type of enzyme that helps speed up reactions by acting as a biological catalyst. So what it does is it helps polymerize DNA. That's why it's called DNA polymerase. So polymerization means to take monomers and make a larger molecule. So the monomers in this case is a nucleotide, and it's going to take those nucleotides and add them to make a polymer of DNA. So here is an unwound DNA for simplistic purposes, and here are the steps. So DNA replication is the process of copying DNA. It happens during interphase, which is S phase specifically when DNA is copied. So what happens is DNA helicase unzips the two DNA strands and breaks the hydrogen bonds. You can see my pair of scissors here is acting like a helicase. It's going to unzip and unwind the DNA. And unzip means to break the hydrogen bonds between the bases, nitrogenous bases. The next thing happens is the DNA polymerase will then match up free-floating nucleotides to template strands. So this DNA opens up. So you can see here it opens up, and the free-floating nucleotides are added via DNA polymerase. And DNA polymerase enzyme knows A with T, G with C. It's actually pretty good at its job. So it's nice. So it's going to add in the bases, hydrogen bonds will form, and then the glue here is just showing that those hydrogen bonds are forming to create a newly made DNA. Now we actually have two newly made DNAs, right? We opened up the original template strand and added in new bases based on the pre-existing letters or nitrogenous bases. So we have two identical copies to the original strand. Half of the strand is old from the original template, half of it's new. This is called semi-conservative, when your DNA is half old or original and half new from the DNA polymerase adding in nucleotides. So you can see the original DNA on the left looks identical to the newly formed DNAs on the right. The color is just supposed to show you where the bases came from. So the greenish color is supposed to show you that it was from the original template strand, and the yellow is supposed to show you that we got those nucleotides free floating from the nucleus. Either way, we're still making the same strands, just more of them. So how accurate is this process? So if there is a mutation is a change in the original DNA sequence because of DNA polymerase didn't add in the correct nucleotides. So you can see our two strands here of DNA, one left side and then right side. And as you can see, a C was added when it should have been a T. So mutations can be corrected. So there is an enzyme that proofreads the DNA and checks for errors and corrects them. So pre-repair, you have a 1 in 10,000 chance that nucleotide has an error, but after proofreading and checking for correctness and fixing it, you have a 1 in 1 billion nucleotide error rate, so not very common. But there's a lot of bases, so there's bound to be mutations somewhere. As long as the mutations aren't lethal, you're in good shape. Here's the practice quiz. Name 3 parts of a DNA nucleotide. Name the 4 nitrogenous bases and how they pair with others. Fill in the DNA nucleotides that are missing, A with T, G with C. Describe what takes place during the 3 stages of DNA replication. Hopefully you think about step 1 being involved with helicase, step 2 being involved with DNA polymerase, and step 3 winding everything up and having your result. And then number 5 is how many mutations are in the DNA molecule below. So you would go ahead and analyze that and point out the mutations. Okay, so this is the next topic that I will stop here and I will go over with you when I return. Thank you.